Hello everyone. Okay, so in this video I'm going to be talking to you about lipid digestion and absorption, so fat digestion and absorption. And quite similarly to the little video that you probably watched recently on protein digestion, there are quite a lot of new words. And you might feel like I am making a lot of this stuff up to be cruel, but unfortunately there are just a few new words in this topic, and you just have to make sure you take your time to get your head around it. And the process of lipid digestion and absorption is a little bit longer than the process of protein or carbohydrate digestion and absorption, so let's just take a little bit longer to get our heads around it. So let's first just remind ourselves about the structure of lipids. So we know that triglycerides are made up of a glycerol molecule and 3 fatty acid molecules, and we'll just draw a very simple diagram to remind ourselves of that. So you eat some fat, nom 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 nom. Maybe you have some nice cake, or you eat some cheese, or you have just, I don't know, maybe you just drank some olive oil. Anyway, you've eaten some fat. When the fat goes through your mouth and your stomach, it doesn't undergo any digestion at all. Lipid digestion begins in your small intestine. And lipid digestion is a mix of both physical and chemical digestion. So one of the first things that we have to do in lipid digestion is we have to increase the surface area of the lipid droplets. Increasing the surface area will increase the rate of action of the enzyme lipase to break the ester bonds, to actually chemically break it down. But first of all, we need to increase the surface area to increase the action of the enzymes. And to do that, we need bile salts. So you've been digesting your food in the stomach for about 8 hours. The partially digested food. Now, the scientific word for partially digested food is chyme. So the partially digested chyme travels through the opened pyloric sphincter into the small intestine. And we already talked in the protein digestion video about how bicarbonate ions that are made in the pancreas are released into the top of the small intestine, the duodenum, and this neutralizes the acidic chyme. So neutralizing the chyme, it was acidic because of the hydrochloric acid in the stomach. It becomes neutralized. This both denatures pepsin, which was the enzyme that was digesting the proteins in the stomach, but it also creates the optimum conditions, the optimum pH for the action of the enzymes that we will find in the small intestine. So we've got the bicarbonate ions that were made in the pancreas, they get released and they neutralize the acidic chyme, and then comes the bile. The bile is made in the liver. It is then stored in the gallbladder and it is released via the bile duct into the duodenum. So it might help us just to have a little bit of context for all of those terms I've just introduced you to. So let's just take a look here. So we can see here is the liver. The liver is massive and lobed, and it's our largest internal organ. So it's in the liver that we make the bile. The liver then moves the bile into the gallbladder, and the bile is stored in the gallbladder. The bile then moves via the bile duct down into the duodenum. At about the same time, we've got the pancreas, and the pancreas is producing the pancreatic juices, which contain digestive enzymes and bicarbonate ions. These are also released into the duodenum, and they neutralize the acidic chyme. And when the bile salts then come into contact with the globules of fat, they emulsify these fat droplets. When you emulsify something, what that means is you break it down into smaller droplets. So let's just have a think about how that works. So let's imagine we have got our droplet of fat, and then it comes into contact with bile salts. Now, bile salts have are polar. They have a hydrophilic and a hydrophobic side. So let's just have a look at that for a second. So fat forms large globules. This is because it doesn't dissolve in water. It hates water. So when it comes into contact with the bile salts, the fat is actually able to form much, much smaller droplets because it is protected by the bile salt, and the hydrophobic side of the bile salt faces the fat, and the hydrophilic side of the bile salt faces the water. So you get these tiny emulsified droplets of fat. So this emulsification by the bile salts of the fat into small globules is important because it increases the surface area for the action of the lipase enzyme, and it means that it increases the rate of digestion. These small globules of fat are now called micelles. So that is a new word that you need to learn. So now we've got these small droplets of fat, the lipase can get involved. Now, lipase is produced in the pancreas, and it is released via the pancreatic duct, and it breaks down fats into fatty acids and monoglycerides, and it does this by breaking the ester bond. So let's just have a look up here. So it breaks it down into fatty acids and monoglycerides. Now, a monoglyceride is a single glycerol molecule and one fatty acid. So if we just think about that here, this is a monoglyceride. So now our lipase has digested, it has hydrolyzed our triglycerides into monoglycerides and fatty acids. At this point, they remain in association with bile salts in this structure called micelles. So this word micelle is both referring to small globules of fat pre-digestion and post-digestion, post-chemical digestion. And each of these micelles at this point after digestion is between about 4 and 7 nanometers in diameter. Now, these micelles are able to transport the monoglycerides and the fatty acids to the epithelial lining of the ileum. Now, why are the micelles useful? They are useful because they increase the proportion of molecules in contact with the cell membrane. Because otherwise, the fatty acids and the monoglycerides would just clump together again in a massive lump, and only a small proportion of them will come into contact with the cell membrane of the epithelial cells. But these micelles bring them to the cell membrane, so that's one function of them. They transport them to the cell membrane, and the second function of them is they increase the proportion of the molecules which actually come into contact with the cell membrane, therefore increasing the rate of absorption. So let's just put this into context of where it is happening. So here we are at the intestinal surface. This is the lumen of our gut. This is the epithelial cell lining our intestines, and we'll say our blood streams down here. And if we remember from before, where are we? These are the villi lining our small intestine. This is the epithelial cell. And then we've got our capillaries. And we also have our lacteals. So the micelle transports the monoglycerides and the fatty acids towards the cell membrane of the epithelial cell. And when it arrives at the cell membrane, the micelle breaks down, and as it breaks down, it releases the monoglycerides and the fatty acids. And we know that monoglycerides and fatty acids are non-polar. This means that they can diffuse directly via simple diffusion across the cell membrane into the epithelial cell. Once they get into the epithelial cell, they are transported to the endoplasmic reticulum. At the endoplasmic reticulum, they are recombined to form triglycerides, and then they are sent to the Golgi body. When they get to the Golgi body, they are there associated with cholesterol and lipoproteins to form chylomicrons. Chylomicrons are specialized structures which are adapted to transport lipids in the lymphatic system. So I know it sounds like I have made up a number of these words, but they are not made up. So new terms you need to know from this bit. Cholesterol, that's not a new word, you already know the word cholesterol. Lipoproteins, you might already know this from GCSE when you learnt about LDLs and HDLs in connection with cholesterol. But the really new word is chylomicrons. So chylomicrons are the form that fats take once they have been recombined into triglycerides at the endoplasmic reticulum and then associated with cholesterol and lipoproteins at the Golgi body. So now they are ready to enter the lymphatic system. So let's just go back to where we are at. So let's imagine that this is our chylomicron that gets popped out of our Golgi body. The chylomicron is contained within a vesicle. And that vesicle transports it towards the cell membrane of the epithelial cell, and where it fuses with the cell membrane of the epithelial cell, and the chylomicron is exocytosed. And where is it exocytosed to? Well, it is exocytosed into the lacteal. So this is where we are. So here is our chylomicron. It was in the cell, then it got exocytosed out, and it enters the lacteal. It enters the lacteal, and then it travels into the lymphatic system. Let's just take a minute to contextualize that. So your lymphatic system is a system of open-ended tubes that run throughout your whole body and perform a number of functions. So take a look at this image here. The green lines are showing you the lymphatic vessel. So this is your lymphatic system. It looks a lot like your bloodstream, but the ends are open-ended. And they're important in controlling the amount of tissue fluid. They're important in our immune system. They're also important in the transport of fats around our body. So let's just take a minute to have a look at our lymphatic system here. And you're probably also noticing our lymph nodes. So can you see these lymph nodes? We have them in our neck, we have them in our armpits, we have them in our groin and in our abdomen. These nodes are really important in our immune system, and when you learn about the immune system, you'll talk about those a little bit more. So let's just have a look at a couple of these lymphatic ducts. So we can see here these open-ended ducts. So we can see here, can you see the right lymphatic duct empties at a junction of the right internal jugular and right subclavian veins. Now, don't worry about all of that. You don't need to know all of that stuff. But what you do need to know is that fats are primarily, so they're first carried in the lymphatic system, and then the lymphatic system drains into our bloodstream. So they carry the fat around, and then the lymphatic system drains these fats into our bloodstream, and then the fats enter the capillaries. So that's what you're seeing here. So where are we so far? We were originally in the lumen of the intestines, then they went into the epithelial cell, then into the lacteal, then into the lymphatic vessel, then they drained into the bloodstream and entered the capillaries. So what happens in the capillaries? So in the capillaries, we are triglycerides again. We started off as triglycerides in the intestine, and we're triglycerides again in the capillaries. So at this point, it looks like we haven't really done any digestion at all. We broke them down, we built them back up again, and then we transported them around our body in the form of triglycerides. Now, in the capillaries, there are enzymes embedded in the membranes of the endothelial cells of the capillary. So if we think for a moment about a capillary wall, so what did we just see? Embedded in the cell surface membrane of the endothelial cells of the capillary are lipase enzymes. These lipase enzymes hydrolyze the triglycerides into monoglycerides and fatty acids. These are then able to diffuse via simple diffusion across the endothelial cell and into the tissues, and then into the body cells. And then they can be used to build up new things like hormones and cell membranes and all sorts of different things that you need that make fat, or they can be stored in adipose tissue as fat. So that was a lot of stuff. That was a lot of new words. That was a lot of steps to that process. So what do I want you to do? I want you to go back and first check that you understand all of the new terms. Make sure you can write down definitions for each. Write down each of the processes. So the emulsification of the fats by the bile salts, and then the transport and the breakdown of the micelles, the activity of the lipase, what happens in the epithelial cell to form the chylomicron, the exocytosis of the chylomicron, and then how the chylomicron gets to the capillaries, and then what happens in the capillaries, and then how the broken down fats, the monoglycerides and the fatty acid, actually get to our body cells. And it's also important that you understand what they can then be used for. So take your time, draw out the process, think about each stage. Also think about which parts of each stage require ATP. But that was a lot of listening, that was a lot of learning, so well done for now.